So I think we got cut off on the last slide, so I will just repeat what I said. So I think this is what we're going to talk about this year in gene technology, which is that before you start this course, you might think that problems are yes or no. Yes, I should do it this way, or no, I shouldn't do it this way. And then my mom thinks that we can actually take pieces of DNA and see them and figure out how to put them together. And we can't do that either. We have to imagine how we see pieces of DNA, and in some cases many, many, many pieces of DNA, and how they all work together. So by the end of this course, we hope that you're closer to this last picture, which is that you understand the subtleties in choosing between different approaches. So one problem that you want to solve could be solved in different ways, and some of those are going to be expensive, and some of those are not going to be expensive, but take a longer time. So trying to balance all of these things. You guys are engineering students, so solving problems in different ways should be something that you're excited to do, I hope. Okay, so that's the end of the introduction, and now we go to the first lecture, which means actual gene technology. So for the textbook, the information about the textbook is also on Canvas. And for this lecture today, we're talking about the chapters 1 and 2, as well as 9, which seems a bit odd, but chapter 9 is really one of the fundamental techniques that you need to know to understand all the other lectures, so we thought it was best just to put it in the first lecture. Okay, so in this lecture, I hope to explain why DNA analysis and gene cloning are important, and give you a theoretical background for understanding how we can manipulate DNA. So not picking it up with tweezers and sticking it together, but the tools that we have in our gene technology toolbox. We're going to talk about how to purify DNA from living cells and how to get DNA without using living cells. For example, vectors and the polymerase chain reaction. Okay, wait, what is DNA? Now I know you guys have had cell biology, so you have some basic understanding, but this course is about going into the details of how to use these techniques. And this is still one of my favorite cartoons. So shortly before discovering the structure of DNA, Watson and Crick, depressed by their lack of progress, visit the local pub. And one of their colleagues, who is sitting a little ways away, says, "I'll have a double, Felix." And they go, "Of course, it must be a double helix and not a single molecule." So I like to start by teaching from the very basic structures of DNA, and the idea is a little bit that if you understand this molecule, it becomes almost like Lego. You can just decide which pieces do you put together to get the structure that you want. And DNA analysis is important because we are reading the book of life, and I think, as many of you also know, there's a lot of talk in the media right now about sequencing the coronavirus. These are also ways that we can track epidemics, we can find out who's infected with what, and we can start to see patterns in how life is assembled. It tells us who we are, and it also tells us how to build ourselves, which is kind of cool or scary, depending on your perspective. And it tells us who other people, plants, bacteria, etc. are, and how they are built. And in fact, we can even start to do really fancy things, like we can sequence all the bacteria on our phone and match it to the person who owns this phone, which, depending on your, again, perspective, is either really cool or really kind of gross. Okay. But before there was gene technology, there were genes and biology. So a lot of the tools that we have in our toolbox, how we manipulate DNA and how we work with DNA, are completely based on how nature also does it in real systems. So we haven't invented anything. We've just kind of taken different systems that are happening in our cells that you may have heard about already in your other courses, and we've harnessed them and put them into different combinations in the lab. So now I need a pointer. There we go. So for example, if you have a regular DNA replication, you have your parent strand, parent or template strand, and there are proteins within the cell that are able to open up this strand. And then this allows for the DNA to become replicated. Then you have small pieces of DNA that are synthesized by another protein that allow later on other DNA proteins to come in and synthesize new DNA. And in the end, you have something called DNA ligase, which allows DNA to be stuck together. So all of these things have been discovered in the context of natural cells and natural molecular biology studies. And now we start to take them in and use them in technology. So you have the whole basic concept of DNA is that you have the template, this up and you can copy it, and then you get 2 copies. This will come again and again in this course. So how this actually happens, it's very important to realize that it only happens at one end of the DNA strand. So you have your DNA strand here, which goes from 5' to 3', consisting of sugar backbones, and then the bases. I think that dinging is just my computer. I'm probably getting a lot of emails. Great. Thymine, adenine, guanidine, etc. And then you have your other strand, which is going from 5' to 3'. And why is that important? Because these are not symmetrical. They're actually in opposite orientation, which means that on the 3' end, you have an OH group here, which if you look on the 5' end, you have a phosphate group. Okay? And this oxygen is able to attack this phosphate molecule. This is organic chemistry, so we don't go into that here. And then this is released, and now this sugar becomes attached in the chain. Okay, so this is a fundamental property of DNA, which allows it to replicate only from one direction, and it also explains why all of our primers need to be written in 5', 3'. We will talk about this more later today. But the key thing to remember is that the end of a DNA molecule that's going to be replicated must be a 3', because you need the OH group with the negative charge on the oxygen. Okay? So, this is all the basics. So, if you understand no other lectures, no, of course you will understand other lectures, but these are the key building concepts. So, another thing that's really important to understand is that the thing that holds these, once you have these 2 strands, the thing that holds them together are hydrogen bonds. Have you guys taken organic chemistry? I can't remember. Okay, probably you have. So you know about van der Waals forces and all these kind of bonds. So hydrogen bonds actually have more energy when they're between the C and the G than when they're between the A and the T. And what this means is that the C and the G's actually hold each other tighter than the A's and the T's. And this means that you can use different temperatures to separate different DNA sequences. So if you look at the 3 sequences here on the bottom, this one has a lot of C's and G's. So those 2 pieces, the 2 sides of the DNA are really stuck together. The ones in the middle are all A's and T's, so they're going to be able to come apart at really low temperatures. But this sequence is the same as this sequence, but this one has magnesium ions. And if you think about charges, the backbone of these DNA molecules is negatively charged. So if you add a positively charged magnesium, it will actually stabilize that structure in a way that's independent of just the A's and the T's. Okay, so you have these 2 sides of DNA, they stick together based on whether they're C's or G's or A's or T's. But then it also is influenced by the surroundings of that DNA molecule. So, a note about writing DNA sequences. So this is a classic question on the exam, possibly on your exam, possibly on someone else's exam, and that is that you're asked to write a DNA sequence that will code for some particular protein. So the convention is that DNA sequences are always written 5' to 3', and this is just a rule. So if you see a DNA sequence and it doesn't have any 5 or 3's on it, we assume that it's 5' to 3'. So that's also something to keep in mind when you work on your assignments or your exam. If you don't put 5' to 3', we assume that that is how you have written it in that direction. So you have, here's your A to 5' to 3'. We'll just remember that the 3' end is where the DNA would be extended from, if there was extension here. But then the other side is going 5' to 3' and would be extended from this end. So if you're going to write this bottom sequence, so this one is pretty easy, 5' to 3'. But to write this sequence into this box, you would actually need to write it A, T, C, G, G. So it would be like this. Because this is the 5' and this is the 3'. So that seems easy enough when this sequence is actually written here. But if that wasn't written there, you would first have to convert and use the opposite base, T's for A's, C's for G's. Then you would have to flip it around and write it in the other direction. Okay. I'm not sure what happened there. What happened there? Yeah, that's just me trying to fill in the box with the computer. But the letters are the same, but the orientation is different. This is another sequence. Okay, good. That's the end of the section about the basics of DNA sequences.